In this video, we're going to talk a little bit about some of the unusual uh, things in regards to microorganisms and how they grow. We're used to thinking about, for us, we eat food, we process that food, and that's one of the things that helps us grow, especially as children. And as we get older, unfortunately, sometimes it helps us grow in the wrong direction. So we're going to talk a little bit about that. On this intro slide, I put some rather interesting microorganisms, and I'm representing growth here. This particular organism is something called Proteus vulgaris. It grows in these little wave patterns, and it can cover this plate sometimes within one day. So it can grow very rapidly. The middle picture is Legionella, an organism you came across in the emerging pathogens that has this characteristic of fluorescing. And that's one way we can diagnose it. And then this last one is E. coli growing on a particular agar that indicates that it's using a particular carbohydrate called lactose. And with the various things that we have in it, it gives you this really neat green sheen that we see here. So we can classify microorganisms and really all organisms by what energy source that they use. An example being a phototroph. If you look at the word photo, as in photon, that's an indication that they're going to be using light, and it's usually sunlight that they use for energy. And we think of this as being plants and algae and certain bacteria that are able to use sunlight for energy. If they're not a phototroph, they're going to be a chemotroph, which means they're going to be using chemical compounds, and that could mean chewing up a dead organism and getting energy from the various components in them. It could be they're eating sugar or something like that. That's a chemical compound, and that's how they're getting their energy. We're going to find that pathogens fall into this category of chemotrophs. And of course, not just pathogens. We are chemotrophs. A cat's a chemotroph, a dog's a chemotroph, that sort of thing. Good bacteria happen to also be chemotrophs. Some of them are not chemotrophs. We do find bacteria that are able to use sunlight as a source of energy, and they're phototrophs. So we find bacteria in both groups. We can also classify organisms by what their carbon source is. An example being, we have something that's called an autotroph. And they can be referred to as self-feeders. The word auto oftentimes means self in Latin. And what that means is they're not depending on anything living for carbon. They're getting their carbon dioxide usually from they're getting their carbon from something like carbon dioxide, that sort of thing, or other inorganic molecules that contain carbon. Heterotrophs, hetero meaning different, meaning they are relying on something different for their carbon source. And so you could say they're depending on other life forms. Either they're eating the life form while it's still alive, or it's no longer alive. If you want to think about it, we eat plant material. That would be considered another life form, and we're getting our carbon from them. We're going to get, if you eat meat, you get carbon from that. So they're using organic molecules. So that's the big difference. Inorganic molecules indicating that it's not alive for the autotrophs, and for the heterotrophs, it's utilizing organic molecules. And they can get that carbon from sugars, from proteins, for lipids, anything that contains carbon. We are going to find pathogens in this particular category. And so here we have a picture of our wonderful cat here. It says, just found out I'm a heterotroph, go figures. And I just added that because I like cats. Now, we never do anything simply in science. We combine those terms to describe organisms in manners of which describe what energy and carbon. So combine those two together, and if we're looking at a pathogen, the energy source for a pathogen, we would use the term chemo, that prefix. If we're talking about what source of carbon they're using, the pathogens would be hetero. And so we can combine those two and get what's called a chemoheterotroph. So I have a picture of a cat here. Yes, cats are, they're not pathogenic, but they are chemoheterotrophs. We are chemoheterotrophs, and many bacteria are chemoheterotrophs, especially pathogens, because pathogens are using us as a carbon source and as an energy source. So other terms that we can use to describe chemoheterotrophs can be parasitic. So if you think about it, that's an organism that would infect you and use you for both your carbon and your energy, but it's not a benefit to you at all. And so it's at our expense. And then a worm would be an example, and I threw in a couple of other cartoons about parasitic things here for you. And then another term is something that's called saprobic. And these are organisms that aren't parasitic, but they are breaking down material. And that's usually the kind of an organism that's going to be breaking down dead material and then taking the carbon and hydrogen and other nutrients from that and using it some for themselves and other it's releasing into the environment. So an example of saprobes, as I've indicated here, they're decomposers. You can think of a lot of the fungi. If you were to go out in a forest and you see that a tree has fallen down, you'll actually see mushrooms growing on them in some cases. And what you can't see necessarily are microorganisms that are also breaking them down as well. And here what they're showing you is kind of what saprobes do. These particular organisms, they can't necessarily get this bigger material that's out in the environment into themselves as a source because their cell wall acts as a really good barrier against it, but they want the material. So what they do is they produce these enzymes that can break down these bigger molecules into the parts that make it up. So if you can break up a protein into amino acids, and then those amino acids are much smaller and they're able to bring that in. And so that's what they do. And we especially see that with fungi. If you were to get spores from a fungi onto something like a blood agar plate, which has red blood cells in it, usually from a sheep, it will release some of these digestive enzymes, and then you get this zone around the fungal colony that's growing where it has destroyed all the red blood cells and taken up the nutrients, so it's clear around it. It's really kind of neat looking. But that's what saprobes are. So if you want to take a look at the whole big scheme of thing with microorganisms, we can break them up into either autotrophs or heterotrophs, and then that's breaking them up by the carbon source, either inorganic or organic. And then you can add to it whether they're using light as an energy source or if they're using chemical compounds as their energy source. So we do have photoautotrophs, chemoautotrophs, photoheterotrophs, and chemoheterotrophs. With this class having a focus a fair amount on pathogens, what we're really going to be looking at a lot is going to be chemoheterotrophs. And so you can see here it's most bacteria, all protozoans, all fungi, all animals, that sort of thing.